Welcome to AP Biology. I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. For an overview of our lesson today, we'll be talking about facilitated diffusion and compartmentalization and its origin. And by the end of this lesson, you should be able to score points for these learning objectives. Go ahead and grab pen and paper if you haven't already. We're going to explain how the structure of a molecule affects its ability to pass through the plasma membrane. We're going to describe the membrane-bound structures of the eukaryotic cell. We're going to explain how internal membranes and membrane-bound organelles contribute to compartmentalization of eukaryotic cell functions. We're going to describe similarities and differences in compartmentalization between prokaryotic and eukaryotic cells. And we're going to describe the relationship between the functions of endosymbiotic organelles and their free-living ancestral counterparts. Before we get started, I wanted to review a diagram that I had in this. It was Fridays, and it had to do with cell membranes and proteins in cell membranes. And I had put together an image like 3 different times during the day, and at the last minute actually put the wrong one in. And so I believe it was only this particular word, the integral, that I wanted to, that I think that it said peripheral on Friday, on the last lesson. Anyway, so I just wanted to take you through this one more time. If we have a transmembrane, okay, transmembranes means we have a protein that spans the plasma membrane, and it's on the exterior and it's located on the interior. So this would be one, and this of course is one, and we see this one. So those are transmembrane. Now this can also be called an integral because it is embedded into the cell membrane itself. So this also could be an integral, but it is not transmembrane. So all transmembrane proteins within the cell membrane are integral, but not all integral are transmembrane. I think I tried to say that in the last lesson, and I'm not sure if I did. Let's go ahead and talk about some other things while we're here. Let's do cholesterol. So here we have our 4 fused rings. There's our cholesterol molecules. And then we have our carbohydrate chains. This particular one is a glycolipid, so that's the glyco part, the carbohydrate part, and it's tethered to a lipid within the cell membrane, so that's a glycolipid. And then we have another one. Here is a, excuse me, we have a glycoprotein, and it's attached to this protein. So there's the glyco part is the carbohydrate part, and then the protein is underneath it, excuse me. And then I think we covered everything, and you know the phospholipid bilayer. Anyway, so in the future, well, we'll go backwards because we're going to put all of them on. There's going to be a Google Drive that you can access that are going to have all the slide decks of all of these presentations. And so what I was thinking is that I know that sometimes my students like to fill in the blanks of trying to remember what something looked like. So I think when it's appropriate, then I'll put a blank slide like this one. Excuse me for being so scratchy, I'm trying. Okay, so like you would say, this was cholesterol, okay? So I'm gonna put in, I'll leave this blank for you, and then when you can download the slide deck, or you can look at this again, you can fill it out yourself. Okay. So now for our warm up, now that you're really ready. Okay, so this was the Friday, the last lesson, which was from membrane transport. And that was the homework then, so it's the warm up now. Proteins, large complex molecules, are major building blocks of all living organisms. And your job is to discuss the following in relation to proteins. First, the roles of proteins in membrane structure and the transport. So there's 2 things you're going to discuss. And I would discuss kind of as a combination of describe and explain. Like the characteristics are important and how and why, if that's appropriate. I'll give you just a moment to work on that. If you already have that ready, then just hold on tight and I'll show you the scoring guidelines. I'm going to get my timer out. Okay. Let's take about 10 more seconds. Okay, so let's look at the scoring guideline. So there are a number of things you could have said, but I want to focus on this is the role and then about the transport. Now this, you could have said a number of things, description of integral and peripheral proteins. I probably would have said that. Membrane synthesis defines membrane sidedness. This, you could have also went into receptors, enzymes, cell-to-cell communication, all of those things that you already know because you've already covered all the content and that you're reviewing now. Role in transport proteins may be specific. Some are going to need ATP, remember those active transport ones. And description of transport mechanisms, the binding, the conformational change, and the release of a molecule. You could have went on into that. And then how proteins form channels. I probably would talk about channels and moving through them. And then you could have elaborated to specifics. You could have talked about ATP synthase and how the hydrogen ion concentration gradient drives the, the production of ATP. And you could have also talked about the sodium-potassium pump, which we will talk about today. We talked about receptor mediated on Friday. All right, I'm going to go ahead and move to the next slide. All right, so what you need to know. So membrane proteins are required for facilitated diffusion of charged and large polar molecules through a membrane. So examples that we're going to talk about aquaporins, and we're going to talk about channel proteins. So as we talked about last time we were together, we talked about how very few water molecules can go through the membrane because they're partially charged. So you must have aquaporins enabled to go from a high concentration gradient to a low concentration gradient of water, okay? Or you could say high free water to low free water, or you could say high water potential to low water potential. Let's see, let's go through here. So we have water can move through this, and it would move towards this area if we had a higher solute in this particular area, in this cell, inside the cell versus outside the cell. Okay, so now I need to make myself to stay focused on this diagram because I want to go into so much more that I will on the next slide. All right, so charged ions, including sodium and potassium, require channel proteins to move through the membrane. So if you look here, it says sodium's high on the outside of the cell. So we have a high concentration of sodium, and it's going to move to a lower concentration of sodium. So it's going to move from high to low, and it's going to it can only do that through a sodium channel. Now, there's a higher concentration of potassium on the inside, and it's going to leak from a high to a low concentration as well. And it also is going to need this channel to do so. Now, once you get the ions on the wrong side of the membrane, when you have the sodium, too much is over here, and then potassium, too much is out here, then you're going to need your sodium-potassium pump to move the sodium back out and the potassium back in. So I'll take that a little bit further on our next image. Okay, membranes may become polarized by movement of ions across the membrane. So I'm going to draw real quickly. All right, so this is a phospholipid bilayer, my version. And let's say we have here's our sodium channel, and here's our potassium channel. And I'll leave it at that. Okay, and then I'll catch the sodium-potassium pump on the next slide. So inside the cell, let's say this is a neuron. The inside of the cell's membrane potential is -70 millivolts. So this is at resting. So this is right here on this graph. Now, if you have something that happens, say it's a neurotransmitter binds or a this is a voltage gated, or it could be a ligand gated depending on its location. And it's going to, when there's an electrical disturbance, this is going to open up. So we have, what we did have was a bunch of sodium out here, right? So the sodium is going to move when it opens from high to low. Now that moment right there takes us from a resting membrane potential of -70 to a +30-ish, okay? So now we're positive on the inside the cell. And then the next channel, this potassium channel, is voltage gated. And when this change in membrane potential happens, then it's going to open, and then the potassium that was higher on the inside is then going to move out of the cell. So that moves us back to negative, but as we'll talk about, and that was this moment right here of heading back. On the next slide, we're going to talk about why that just can't be okay. All right, so the sodium-potassium pump is going to contribute to this. So what I just did was take us through here in step 1, we have higher sodium on the outside and low sodium on the inside. We have high potassium on the inside and low potassium on the outside. And this is normal resting potential right here, okay? So if we, let me get the color. Now, if this channel was, the sodium channel was to open, then that sodium is going to move through. And that's going to change the membrane potential then to positive. And that's where that +30 mark is coming in, 20-something 30. Okay, so now we have more sodium and more potassium on the inside. So it is positive. And now the potassium channel is going to open. Where are we doing that? Okay, so we're in. All right, we have to do it here. Potassium is going to open, and then the potassium will move, here we go, to the outside. So we have had an increase on the inside potassium, and it went through, and now we have the potassiums on the outside. So we're back to a negative membrane potential, which is fine, right? But another action potential can't happen if you have the wrong ions in the wrong place. You can't have your sodium on the inside, your potassium on the outside. So the sodium-potassium pump is going to maintain that membrane potential and allow more action potential to occur by getting them in their right location. It's going to pump the potassium back in, and it's going to pump the sodium back out. Okay. All right, I think I've showed this image so much, and it's going to continue on because we're going to be going into photosynthesis and cellular respiration for the next few days. So just at least you know this diagram. All right. Membrane proteins are necessary for active transport. Metabolic energy, such as from ATP, is required for active transport of molecules and or across the membrane and to establish and maintain concentration gradients. So let's look at this gray area. All right, so here's our gray areas. And remember, this was areas of higher concentration. So we have a higher concentration of hydrogen ions built in this intermembrane space and also in these thylakoid lumens. So why? So it's being compartmentalized in that location to establish this gradient. So it's going to be for a purpose in photosynthesis and cellular respiration. But how does this happen in the first place? It's going, if we're pushing hydrogen across a membrane, we're going to have to go from a low to a high concentration gradient. And to do that, we're going to need to use ATP. So from our last lesson, this was active transport, right? We went from a a low to a high concentration, and we needed ATP to do that, okay? From low to high. Okay, so in my classroom, you probably wonder what blocks have to do with this. Before I allow kids to try to measure Jell-O with a ruler, I have them practice with blocks, and I have them determine the surface area and the volume, and then get their ratios and learn all the math. This year we added a cylinder, okay? And so because that in the CED, there was the surface area of a cylinder and a volume of a cylinder was added. So after we spend our time and we know what we're doing, then I let them loose on the jello. So investigation 4, part 1, diffusion. To maintain homeostasis, materials move in and out of cells. Diffusion moves these materials, these molecules, from an area of high concentration to low concentration. This lab uses artificial cells to help students study the surface area to volume relationship in regards to diffusion efficiency. We first learn how to calculate surface area and volume, and then how to present the ratio. The cubes are made with unflavored gelatin, sodium hydroxide, ethanol, and phenolphthalein. Then the cubes are placed in vinegar and timed until they turn clear. Why do the cubes turn clear? The vinegar and acid diffuses into the basic block, decreasing the alkalinity. However, the indicator that turned the blocks pink was in a response to an alkalinity, and that's why they're turning back clear. Okay, so here's a sample of my kids. There's their blocks. Okay, those are really huge blocks, and they take a very long time to get some results with blocks that large. All right, so here's a sample, a good sample. We have a small sample. These are kind of out of order. You can see that this one is already clear, and this block is almost clear, and this one has quite a ways to go. So here's a sample. Oh, wait. So this, I can't show this, but it is linked to into the PowerPoint slide that will be available to you if you wanted to watch it. It's on my YouTube channel. But this was it's a, what do you call it? A time lapse. Kids do that in my room a lot, and they'll to show their experimental process along the way. So that's the small block. This was the medium block, and then way back here was the larger block. And so this one went clear first, and then second, and then third. Okay. All right, and here's some sample results from doing so. Here's cube 1, cube 2, and cube 3. And I recommend that when you're trying to, and if you had this as a test question on an exam, that you put your chart in the order that you need to say it. Like if you need to, at the end, if you need to talk about the surface area to volume, then make sure you have, here's the surface area, here's the volume, I divide, and then I get this, okay? So cube 1, we had a surface area to volume of 1.5 to 1, in cube 2, 3.3 to 1, and then in cube 3, 6 to 1. So the smaller cube, here we go, smaller cube right here, this was a medium cube, and this was a large cube, had a greater surface area to volume ratio, and it went clear the fastest. This meant that the if this was a cell, the nutrients can diffuse more efficiently into this smaller cell. All right, compartmentalization. Eukaryotic cells maintain internal membranes that partition the cell into specialized regions. Intracellular metabolic processes and specific enzymatic reactions, I'm going to go ahead and underline that, are compartmentalized within membrane-bound organelles and eukaryotic cells. This compartmentalization contributes to the functions within a eukaryotic cell. Okay, so we have all of this is happening. We're establishing these concentration gradients, and we're compartmentalizing it into intermembrane space here and in the lumen of the thylakoid for a reason, for those enzymatic reactions to be allowed to happen. Okay. All right. Prokaryotes generally lack internal membrane-bound organelles, but have internal regions with specialized functions. And let's draw one real quick. All right, so let's say this is our prokaryote, and it has one, oh, I didn't want to use that color. It has one continuous long circular chromosome, okay? It doesn't have a nucleus. It doesn't have a nuclear membrane-bound nucleus, but what it does have is, it's called a nucleoid region, okay, where you'll find that. All right, so one circular chromosome is located in the nucleoid region of a prokaryotic cell. Other internal regions perform respiration and photosynthesis on extensions of their cell membranes that use a proton gradient and enzymes to produce energy. So you may have, let's see, let me do an example. So say this is a prokaryotic cell with respiratory properties, okay? Let's use that word. Then there are other internal membranes that they can have the respiration occur on. And same thing if this was a photosynthetic. There's other internal membranes where this happens. Okay, theory of endosymbiosis. I know I've already talked about this in one of the evolution videos. Membrane-bound organelles evolved from once free-living prokaryotic cells via endosymbiosis. So here we have our prokaryotic cell right here. And here is our circular chromosome in our nucleoid region. And in the next step, we have that cell membrane is invaginating into the cell. And this will eventually be our nucleus. And this will then have your endoplasmic reticulum and your Golgi and your vesicles and your plasma membrane. All that's made of the same materials, and that would be part of what's called the endomembrane system. Different from endosymbiosis, but sounds a lot alike. Okay, so in the next stage, you see that this large cell is engulfing this prokaryote that has respiratory properties, okay? So now this cell is using this prokaryote to produce ATP, okay? And the same thing can happen with the chloroplast. This was originally a prokaryotic organism, came in with membranes that had photosynthetic functions, and then it became a chloroplast, okay? And we know this because two major reasons we know this is that the double membrane, so say this was a cell that is engulfing this other cell, see the membrane of the cell one engulfing the other cell, and that makes us have our double membrane. Another feature is that this prokaryote came in with its own DNA, and so did this chloroplast. And as mitochondria and chloroplast, well, that as a prokaryote, as a mitochondria and a chloroplast, they have their own circular DNA also. All right, so the science skills that we're going to focus on, 6B, support a claim with evidence from biological principles, concepts, processes, and or data. And then we're going to also predict the causes of effects or of a change in or disruption to one or more components in a biological system based on biological concepts or processes. And some task verbs that we'll use a couple of times today are describe and support. Excuse me. Okay, so we're going to go through about 4 questions, I think. Some multiple choice a little bit later to see if you understand all the content, but we're going to write this one FRQ together. So what I want you to do first is what I do. You don't have to do, but look at the diagram first. Here we have a cell, and I can tell we have a eukaryotic cell because we have a nucleus. We have endoplasmic reticulum and some Golgi bodies, right? We have mitochondria, but we have also this apicoplast, and that might be a new word for you, but that doesn't necessarily mean anything, okay? You just need to look at the characteristics of this. If you can see, it has its own DNA, and then it has another feature. It has what? It has a double membrane. Okay, now let's read the question. Based on figure 1, describe 2 pieces. Okay, so if I was teaching this in a classroom, I would say, box in your command words, circle your how manys, pieces of what? Of your evidence a researcher could use to support the claim that apicoplast evolved from free-living prokaryotic organisms. I'm going to give you just about 20 seconds to jot those down quickly. Okay, let's look at the scoring guideline. So if you said possesses a double membrane and possesses DNA, you have both of those points. All right, so now let's do the multiple choice to make sure you understand everything that we've talked about. Number 3, researchers claim that a particular organelle originated from a free-living prokaryote cell that was engulfed by a larger cell, as shown in figure 1. So here's figure 1. All of this is figure 1. Here's the question where it starts. Which of the following provides evidence to best support the researcher's claim the organelle has a phospholipid membrane? Remember, we have to support the researcher's claim that said it was engulfed, okay? The organelle has protein in the membrane. C, the organelle has a double membrane. Or D, the organelle has internal aqueous environment that is similar to the cytosol of the larger cell. So 10 seconds there, make your decision. And I hope you said C, right? That is the only one. While other answers can be right, this is the one that answers what they're asking to support the researcher's claim. And then you have to say, what exactly did he claim? He claimed it was engulfed by a larger cell. All right, so this is a familiar diagram. I'm sorry this print is so small. It's the best I could do to get on here, but at least you're familiar with it already. A cell's membrane potential is maintained by the movement of ions into and out of the cell. A model showing the influence of membrane proteins on the movement of sodium and potassium ions across the plasma membrane is presented in figure 1. Okay, so we're familiar with figure 1 already. All right, based on the model presented in figure 1, which of the following outcomes will most likely result from a loss of protein X function? Oh, here we go. Okay, so if we took out protein X, this looks just like what question 2, right? Part C, where you're going to predict what's going to happen if you take something out or you change it. Okay, this is similar. All right, so based on the model in figure 1, which of the following outcomes will most likely result from loss of protein X function? A, the membrane potential will be disrupted by an increase in sodium concentration inside the cell. B, the membrane potential would be disrupted by an increase in K concentration inside the cell. C, the membrane potential will be maintained by the sodium-potassium pump, moving more K ions into the cell. And D, the membrane potential will be maintained by the diffusion of sodium ions in the cell. I'll give you 20 seconds. Okay, let's take a look at the answer. And I hope you chose B. Okay, let's talk about why. All right, so let's take out protein X. So what can't happen now? K is not going to be able to leak out. So it's going to be still in here. This pump, okay, this only has to do with sodium, and this pump is going to pump potassium back in. So this is going to be disrupted because of increased K inside of the cell. All right, which of the following statements best supports the claim that certain organelles within eukaryotic cells evolve from free-living prokaryote cells? The cytoplasm of both eukaryotic and prokaryotic is surrounded by a plasma membrane. Oh, this kind of looks like something we've already did, just a little bit different. Eukaryotes and prokaryotes both contain ribosomes, but the ribosomes of eukaryotes are more complex in structure than those of prokaryotes. C, eukaryotes exchange segments of internal membranes between the endoplasmic reticulum and the Golgi apparatus, but prokaryotes have no such internal membranes. D, some organelles contain their own DNA that is more similar to prokaryotic DNA in structure and function than to eukaryotic DNA. Okay, so just take 5 seconds because I know you know the answer. Okay, and the answer is D. All right, I think that's the end of my multiple choice. No, sorry, just kidding. All right, this is number 11 in particular test bank questions that I made. All right, the endosymbiont theory proposes a model for the evolution of mitochondria. According to the model, an ancestral eukaryote engulfed a small, free-living prokaryotic organism. The engulfed prokaryote then formed an endosymbiotic relationship with the eukaryotic host. Which of the following observations best supports that model? A, prokaryotes and eukaryotes acquire nutrients from the surrounding environment. B, organelles such as mitochondria and the endoplasmic reticulum have membranes composed of phospholipids. C, mitochondria and some prokaryotes share similar metabolic reactions that produce ATP. And D, eukaryotes evolved after prokaryotes and have more complex structures. I'll give you just 5 seconds on that one. And I hope you chose C. Okay, now that's the end of, I think that you pretty much have established that you understand the theory of endosymbiosis. All right, so now we're going to do an FRQ together. I love this question. It actually made me go look for the research and I actually saw there was what I found online that I can't put on here, but I saw these red buds and then the blue flower. The petal color, let's look at the boxes first. So what do we have in our table? We have vacuole pH, we have flower color, and we have cell volume. So in the bud, we have a 6.6 pH, and it's red, and it's small. The cell volume is small, okay, the cell. And then the vacuole pH in here is 7.7, and then it turns blue, and then it's large. So this more alkaline pH causes it to be blue. All right, so what's going on inside the cell? The cells have a potassium channel, and that one has a potassium channel. They both have proton pumps. There's one, there's another. I can see something different here. The potassium hydrogen transport protein is active in the open flower, what it is not in the bud. Okay, and then we see water moving into the vacuole. So let's read this. The petal of the Mexican morning glory changes from red to blue, and the petal cells swell during flower opening. The pigment heavenly blue anthocyanin is found in the vacuole of petal cells. Petal color is determined by the pH of the vacuole. A model of the morning glory petal cell before and after flower opening is shown in table 1. So the first thing you need to do is we're going to identify the cellular component in the model that is responsible for the increase in the pH of the vacuole during flower opening. So we're increasing pH. What is responsible for? Just write the 1, 2 words down, whatever it takes. Okay, go ahead and do that. All right, and then you're going to describe the component. What did it do? Okay, so you identified something, and then why did you say it was that? Is what you should answer there in your describe. What's happening at that location? I'll give you just a couple of seconds, maybe 10. All right, and then part B, a researcher claims that the activation of the potassium hydrogen transport protein causes the vacuole to swell with water. Provide reasoning to support the researcher's claim. So what happened? Because the researcher claimed that this transport protein caused the vacuole to swell. So he said that this right here caused this to swell. Okay, so you describe why. You provide the reasoning why. And I'll give you 10 seconds for that. Okay, so let's look at the scoring guideline. And part A, would have gotten you both of those task verbs. The potassium hydrogen transport protein is identified as a cellular component and then responsible for the increase in pH. So you probably wrote more than it was needed, but it's better to write a complete thought than to try to figure out what the rubric wants you to do specifically. Like, answer the question completely. All right, part B, response includes one of the following reasonings. The concentration of solute K is increasing inside the vacuole, and the solute K is moving into the vacuole, and that's making it hypertonic, hyperosmotic, and lowering the water potential. So you have tomorrow with Ms. Kate, you're going to be reviewing osmosis, water potential, and tonicity. All that will happen tomorrow. But if you figured it out today, that's good, because look at all this K. All this K had higher solute in here, so that water is going to move right in there. Okay. So your takeaways for today: the structure of a molecule affects its ability to pass through the plasma membrane. Membranes contribute to compartmentalization and contribute to the functioning of eukaryotic cells. You should also be able to describe the membrane-bound structures of the eukaryotic cell. You should be able to describe similarities and differences in compartmentalization between prokaryotic and eukaryotic cells, and describe relationship between the functions of endosymbiotic organelles and their free-living ancestral counterparts. And always, my motto: describe it, explain it, and predict what you would do without it. Okay, some common errors and misunderstandings. Students, okay, so if you write this, it is not the same as this, okay? If you are in interpretation wise, if you were to say the same numbers and you were to say 10 to 1, and then you had 10 over here and you said your surface area to volume ratio is 1 to 10, you have to be very careful in how you write things, in what order, and then how you complete your sentence with those terms. All right, and then another place is the location of proton gradients and their role in ATP production. We're moving into photosynthesis and cellular respiration after osmosis tomorrow, and that's really exciting. And I think that we've probably established a real good handle on proton gradients, and then we can just take it one step further and you have photosynthesis and cellular respiration down. Then tomorrow you will talk more about osmosis and all that, but the movement of sodium and potassium into the cell and out of the cell, and how it's getting in one place, and that sodium-potassium pump, you really don't need to know the action potential part of the cell membrane, but it's a good way to learn the role of the sodium-potassium pump in a cell. All right, so for your homework, you need to, okay, so a difference between prokaryotes and eukaryotes is seen in the organization of their genetic material. And your job is to discuss the organization of the genetic material in prokaryotes and eukaryotes. Now, I do not want you to just write the prokaryotes do not have a nucleus, they have a nucleoid region, and the eukaryotes have a nucleus. It's bigger than that. Go back into the things that you learn in, let me give you some hints if I can think of what's on the rubric, in gene regulation. Okay, go into unit 6 and think of things that you could say about this genetic material to be different, prokaryotes versus eukaryotes. Okay, all right, and device and internet access, you still have time. Do this by April 24th. We know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know at cb.org/tech. And I got you out of here in about 35 minutes today. Thanks for watching. We'll hope you'll join us again. Be sure to complete your homework question. Be ready for the beginning of the lesson tomorrow on osmosis, which is membranes osmosis and the osmosis portion of investigation 4. And that's with Ms. Kate tomorrow. Thank you. Bye.